Welcome back to The Explainer. So today we're diving into something really cool. Picture a bright, clean classroom. You've got your notebook open, and I'm right here at the whiteboard ready to map this out for you. We're looking at the evolution of biological classification, but we're going to look at it through a really fun lens. Think of the history of biological classification as a series of massive software updates to our understanding of biology. Over the centuries, scientists have continuously patched, updated, and rebooted the very system we use to organize life on Earth. It really makes you wonder, though, how on Earth did humanity actually figure out how to organize millions of different species of life? I mean, it is a truly monumental task. Just imagine trying to build the ultimate filing system for nature, taking a wildly messy desk covered with millions of completely unique, living items, and trying to sort them all into neat, logical little folders. Sounds impossible, right? Okay, let's dive right into this. Here is our step-by-step roadmap for today. 1. The pioneers of classification. 2. The Two-Kingdom era. 3. Adding a third kingdom. 4. The Five-Kingdom revolution. And finally, 5. The modern three-domain system. Starting right off with Section 1, the pioneers of classification, our early filing system. Now, this human need to categorize the living world around us isn't a new thing. It actually stretches all the way back to ancient times. Let's sketch this out on the timeline. Starting all the way back in the 4th century BC, the Greek philosopher Aristotle first grouped plants and animals. His work was actually later translated into Arabic by Ibn Rushd in 1172. Then, between 781 and 869 AD, the Arab scholar Al-Jahaz meticulously described 350 species of animals, and he even wrote extensively about the complex lives of ants. Incredible, right? By the 16th century, Andrea Casalpina began dividing plants into 15 groups called genera. Fast forward to Tournefort, who introduces the concepts of class and species. And all of this brilliant foundational work perfectly set the stage for Carl Linnaeus in the 1700s. And let me tell you, the Swedish biologist Carl Linnaeus was an absolute game changer here. He stepped up and created a formal taxonomic hierarchy using 6 distinct levels: kingdom, class, order, family, genus, and species. This finally gave science the rigid, universally understood structure it desperately needed to classify life seriously. Which brings us nicely to Section 2, the Two-Kingdom era, or as I like to call it, Taxonomy 1.0. This was the very first formal classification system, the absolute original basic version 1.0 for organizing life. In this earliest system, basically every living thing was just dropped into one of 2 giant buckets: Kingdom Plantae or Kingdom Animalia. The logic was super simple. If it makes its own food, meaning it's an autotroph, it's a plant. If it cannot make its own food and has to eat other things, a heterotroph, it's an animal. At the time, early scientists basically threw literally everything else they found, including bacteria, archaea, and fungi, straight into the plant box. Good enough, right? Well, as our microscopes got better, our worldview got a whole lot wider. So the big question became, what happens when an organism just doesn't fit into the plant or the animal box? Well, you get some major system bugs. Taxonomists very quickly found version 1.0 totally unworkable because of oddities like the euglena problem. See, euglena is this microscopic organism that uses plant-like chlorophyll to make food, but in the dark, it eats like an animal. Plus, it completely lacks a cell wall. On top of all that, this rigid two-kingdom system completely failed to separate simple prokaryotes like bacteria from the more complex eukaryotes. It was definitely time for a patch. So here is Section 3, adding a third kingdom, Taxonomy 2.0. This first major update was driven entirely by the discovery of those microscopic oddities. In 1866, the German zoologist Ernst Haeckel proposed a brilliant workaround, a brand new third kingdom called Protista. He took all those confusing euglena-like organisms, along with bacteria and archaea, and just moved them completely out of the plant kingdom and into this new category. Problem solved, right? Well, not quite. Now, what's really interesting about this is that even with a third kingdom, the system was still breaking down. This time because of fungi. Taxonomists started to realize that mushrooms and molds are absolutely not plants. I mean, they are heterotrophs. They get their food by absorption. Furthermore, they don't even have cellulose in their cell walls like plants do. They possess chitin instead. So the filing system was fundamentally flawed once again. And that brings us to Section 4, the Five-Kingdom Revolution, Taxonomy 3.0.
This is exactly where our growing understanding of cellular structure forces a total, ground-up redesign of the biological framework. But before we could build a better system, we really needed better vocabulary. So in 1937, French biologist E. Chaitin suggested two absolutely crucial terms: prokaryotic, for simple cells like bacteria, and eukaryotic, for complex cells with organized structures, which are found in protista, fungi, plants, and animals. Defining these cellular baselines was the key to unlocking the next massive leap forward.
Enter American ecologist Robert Whittaker in 1967. He stepped up and created a highly logical, step-by-step sorting method. Step 1. Look at the cellular organization. Is it a simple prokaryotic unicellular organism, or is it eukaryotic? Step 2. Look at its mode of nutrition. Does it use photosynthesis, like a plant, absorption, like a fungus, or ingestion, like animal?
Based on those two very clear steps, Whittaker gave us the famous 5 kingdoms: Monera, Protista, fungi, plantae, and Animalia. It was so elegant. Fungi finally had their own kingdom. And it's definitely worth noting here, this system got an amazing software patch of its own in 1988. That's when biologists Margulis and Schwartz modified it to include genetics alongside cellular organization and nutrition. They kept the same 5 kingdoms, but made the sorting criteria incredibly robust.
But science never stops, does it? Let's move to Section 5, the 3-domain system, the modern framework. Because it turns out even the 5 kingdoms weren't the ultimate top level of the hierarchy. In 1977, American microbiologist Carl Woese completely shifted the paradigm. By studying genetics, he uncovered something truly amazing. What we thought was just one big group of simple prokaryotes actually consisted of 2 fundamentally different types of organisms. So to fix this, he didn't just add another kingdom, he added a brand new level of classification entirely above the kingdom level. He called them domains. So the absolutely crucial takeaway here is that all of life is now divided into 3 domains. You have domain archaea and domain bacteria, which split those simple prokaryotes into their rightful separate categories. And then you have domain eukarya. All the eukaryotes, the protista, plantae, fungi, and animalia, are now completely grouped under this one massive eukarya umbrella. And this brilliantly illustrates the whole evolutionary story we just talked through. Over on the far left, you see the simple 2-kingdom system we started with. Then we move into the 3-kingdom system to patch those early holes, followed by Whittaker's incredibly robust 5-kingdom framework. And finally, sitting over on the right, the grand 3-domain umbrella sitting proudly on top, elegantly sorting out the bacteria and archaea from the complex kingdoms beneath them. It really highlights how scientific truth is a constantly evolving process of refinement. Which leaves us with a fascinating thought to close on today. As our gene sequencing technology gets even more advanced and we look deeper and deeper into the microscopic world, what hidden bugs in our current system will we find next? And when we do, what is taxonomy 5.0 going to look like? Thanks for joining me for this explainer. Keep taking those notes, keep asking questions, and I'll catch you next time.